Good afternoon, everybody. Thanks so much for having me virtually to visit Columbia. I hope that you guys in New York have had at least some nice weather like we have in Boston the last week or two to make up for all the other challenging difficulties during these days. I'm not actually going to talk about COVID, but I kind of feel like we all should have a little COVID representation in our talks these days. Thanks especially to Wei Li and colleagues for inviting me. Very sorry that I can't be there in person. Look forward to maybe the spring or next summer to be back in New York and at Columbia with all of my friends and colleagues there. I'm going to be talking today about some of the work from our group over the last 5 to 10 years, focusing on really how we can understand threat processing and fear processing in the brain, mostly focused on amygdala, but certainly some other areas in humans and in animals, and how we can use this to really take us to the next level of understanding new treatments for post-traumatic stress disorder. And this is all really framed in the context of why post-traumatic stress disorder is a particularly interesting and I think academically exciting area to study, on top of it being extremely compelling clinically. It affects about 5 to 10% of the population. It's one of the highest risk disorders in psychiatry for suicide. It's associated with a whole host of comorbidities and other disorders. Those are all clearly significant reasons to study it. But there are many interesting psychiatric disorders. I'm particularly interested in PTSD because I think it's one of the most tractable of the psychiatric disorders. I think it's tractable because it has a shared neural circuitry of fear and threat that we understand across mammals and really in many ways across vertebrates that I'll talk about. We know when it starts. It's one of the few disorders in psychiatry where we actually know the time of it starting. It starts by definition at the time of the trauma. And it involves learning and memory and synaptic plasticity. And these are some of the areas that we understand the best about and have made some of the most progress as a field in the last 20 to 30 years. And so our goal then is to have a molecular neurobiology of PTSD that can focus on predictive biomarkers, leading to novel interventions and novel treatments. So of course, threat and fear are critical evolutionary emotions for survival of all species. What's interesting is with post-traumatic stress disorder, it seems to be a case where the system gets out of control. And so this classic figure from Joe Ledoux, we see what he used to call the low road, where the sensory system, the eyes are picking up. In this case, this guy's walking over a stick, but he might or might not be a snake. The pre-processed information goes to the thalamus, and then there's a direct thalamic projection into the amygdala. In the olfactory system, for example, there's direct olfactory projections into parts of the amygdala, such that it's much faster than most other emotional or reactive brain areas. The amygdala is receiving within a few synapses information from the sensory world. That sensory information from the thalamus is then highly processed in the visual cortex, higher order associative cortex, decision-making areas, and then hundreds of milliseconds later also gets to the amygdala. But in those early milliseconds, the amygdala then pulls a fight or flight trigger, if you will, to activate the survival reflex. And what's again exciting from a research perspective is this region is really conserved across all mammals, and at least a fair amount of the circuit connectivity we're understanding, and more recently single cell sequencing and other types of cellular specificity are also conserved across species. And so then it lets us bring together this idea that fear and threat are things that happen all the time. They're part of the normal continuum of emotion, but dysregulated fear that's out of control is really behind some of our most critical disorders in anxiety and psychiatry, particularly the phobias, panic disorder, and post-traumatic stress disorder. And with PTSD, the increased hypervigilance, intrusive thoughts, re-experiencing and anxiety, the difficulty with concentrating and memory, the difficulty with sleep, and the deficits in extinction and recovery from fear are all things that we can start to really understand at a neural circuit level. And it's really, I think, a fascinating opportunity to both bring together just inherently interesting understanding of how the brain creates behavior with potentially new approaches to helping patients. So from a human perspective, somewhat the canonical / overly simplistic model is that the amygdala is again sort of serving as this Pavlovian area in which previously neutral cues, like a conditioned stimulus, say you're in a traffic accident, the cars, the sights, the sounds, the parts of town, are paired with the specific unconditioned in our mouse lab, that would be an unconditioned shock. In the real world, it's trauma, whether it's a car accident, whether it's an interpersonal assault, or any number of other life-threatening traumas. And that essentially one trial learning is now in place such that in the future, the things that are similar to the conditioned stimulus alone are sufficient to trigger the hardwired outcomes that were consistent with the unconditioned stimulus previously. So that through connections in the lateral, basolateral, and central amygdala, and then hardwired outputs from there, it's now known that a whole host of survival reflexes are activated. And one of the fascinating things that happened as an observer of both neuroscience and psychiatry really through the 80s and 90s was the realization on the one hand of and sort of the identification of panic syndrome and a panic disorder as having a whole set of cardiovascular, GI distress, and physiological responding. And separately, work by people like Mike Davis and Joe Ledoux and Mike Vanzelo and others who were working to really work out the neural circuitry of the amygdala showed that hardwired outputs from the amygdala to many subcortical areas led to the same set of syndromes and specific rapid symptoms: the heart rate changes, the GI distress, the bradycardia, the respiratory distress, the increased arousal and hypervigilance, the increased startle, freezing, and the hypothalamic pituitary adrenal axis stress response. So what that allows is one of the few places in psychiatry where we have essentially a cortical neural reflex, a cerebral, neural reflex, leading to a specific set of threat behaviors that very well maps on to a specific syndrome that we see in humans, the panic response. And while we see the panic response in all sorts of things, and we often associate it with panic disorder, we very much see it in post-traumatic stress disorder. And what's interesting about PTSD is, by definition, PTSD is defined on the instigating incident of CS, unconditioned stimulus pairing. So it's really Pavlovian conditioning sort of borne out. And we now, of course, know that it's not just the amygdala, it's being regulated by the medial prefrontal and dorsal anterior cingulate cortex, the hippocampus, that have different components of fear modulation, threat modulation, and extinction that we'll get to. So how do we think about post-traumatic stress in the context of symptoms, timelines, behaviors, and neural circuitry? This model is one that I think sort of can combine all of those at some different levels. And the first top level question is, we know, given trauma, only about 5 to 10% of the people who experience a life-threatening trauma will go on to develop PTSD. And the vast majority, following a single trauma, will recover. And so what's the difference? So what are the different pre-existing sensitivities? Well, we now know, and I'll show some of the data, that there's genetic heritability. Twin studies and now large-scale GWAS consortia show that 40 to 50% of the differential risk of PTSD is genetically inherited. Of course, you have to experience the trauma. If you grow up in a situation, in a fortunate life that you never experienced severe trauma, you may never know if you have that proclivity or not. Very much as diabetes and certain forms of other eating disorders or obesity, et cetera, can be partly genetic, but if you're not in a situation to eat certain kinds of foods, you may not be associated with that risk, and similarly with substance abuse. We also know environment is critical from a perspective of total trauma exposure. The risk for anyone with a single trauma is 5 to 10%, but if you have multiple additive significant traumas, particularly interpersonal traumas, that risk can be much higher. And those who have significant childhood trauma, they even have a higher risk. And so what that leads to then is really a very interesting case in our disorders where we can have a detailed understanding of the genetics, but also a fairly good understanding of the environmental threats. And so a place to understand how gene and environment interact to predict risk, and how those interactions then alter the different aspects of the circuitry from the perspective of creating the initial consolidation of fear and threat memory that creates the initial PTSD symptoms to those that maintain the symptoms and may prevent recovery. Again, one doesn't have to, it's Coles to Newcastle to say anything about learning and memory at Columbia, but of course memories aren't created initially, rather they're consolidated over the course of minutes to hours to days to even weeks. And so we think there's an early molecular consolidation, a later synaptic consolidation, and a later systems consolidation in terms of brain regions. But that also then allows us to raise the question of if there are ways we could understand and intervene early in the consolidation period, maybe not immediately, but within the first few hours, or maybe just before the first night of sleep in the emergency department we're on the battlefield, could we actually prevent PTSD and have one of the few disorders in our field where in a neurobiologically driven way we could prevent the disorder? And that, I think, is a very exciting thing. And so a lot of the talk today will be on the ideas of consolidation of fear and what happens in those early minutes and hours, and might we be able to manipulate that. Once we're into the weeks following the trauma exposure, people express the symptoms of PTSD, even though officially the first month of symptoms we call acute stress disorder, after 1 month PTSD, after 3 months chronic PTSD. We really think from a biological perspective, it's a continuous syndrome. And that's where you see the PTSD symptoms of intrusive memories, nightmares, flashbacks, avoidance behaviors, increased hyperarousal response, startle response. And we think certain sorts of behaviors are both sine qua non with the disorder as well as reinforcing of the disorder. So, for example, those with PTSD are more likely to generalize memories, and generalization really is defined as recruiting previously non-associated things with that. So for an example, someone might have been attacked down a dark alley and they have bad memories of that, but then they start getting afraid of men in general, and they become afraid of leaving the house at night, and then they don't want to leave their house at all, and then they don't want to leave their bedroom. The context of the world become more and more dangerous and less safe. And that's in contrast to those who recover, who are more likely to discriminate, and they fear is appropriate in the right places and situations, but is not everywhere. There are safe places and there are dangerous places. Alternatively, those who develop PTSD tend to do what historically was clinically called sensitization, which obviously is different than aplesia sensitization, but the concept is similar. Every time you get upset about it, think about it, it seems to get worse. And that seems in contrast to the basic mechanisms of recovery that are very much based on Pavlovian extinction, where there seems to be a diminished response to fearful cues over time. And how we are now thinking about it more is the concept of reconsolidation that Kareem Nader and Joe Ledoux and others have worked on, Susan Surratt, since 2000. Maybe what's happening with the reactivation of a memory, and that all memories, when reactivated, have an ability to be labile again, and that some combination of reconsolidation and strengthening them, or activating new safety circuits that becomes new plasticity dependent inhibitory safety circuits that occurs with extinction, are always pushing and pulling every time a memory is activated. And so when we extinguish a memory in psychotherapy, that requires certain constraints on it that maybe are pushing towards extinction and pushing away from reconsolidation. I'm not going to talk today about reconsolidation and extinction, because I'm really going to talk just on the early stages of consolidation. So my talk's 3 parts. The first 2 are biomarkers of trauma consolidation, talking about some of the early studies and where we are in terms of understanding memory consolidation in PTSD, and of course, mostly from our own work. There's lots of other stuff I won't be able to cover today. Translating this a little bit into cell-based and genomic markers and targets, and where we are with the Psychiatry Genomic Consortium, some of the large-scale multi-ohm site consortia to how large-scale genetics brought to PTSD, and how that overlaps in at least some ways with some things where you're starting to understand more about mice, and how we can take that to the next level. And then really end with how our basic understanding of fear consolidation and amygdala processing can allow us to start targeting molecules, receptors, and cells in more specific ways, both with single cell approaches as well as with identifying new individual pharmacological targets or groups of targets for maybe future tools for intervention. So, of course, the concept of consolidation is one that's been around in different ways for a long time, proposed over 100 years ago by Mueller and Pilitzker, and Jim McGaw in the human behavior and rodent behavior space did a lot of work in this over many decades, really helping us in modern world to better understand short-term to long-term memory, and that these really through multiple ethics, short to long-lasting memories, and that those memory phases have different molecular and circuit constraints. Obviously, most of what we understand molecularly about learning and memory originally bore out of the Candel and Schwartz and other labs out of Columbia, and it still remains fascinating that so many of the molecules and pathways related to plasticity came out of some of the early work in aplesia and models. And I'll actually be showing some new data today on creb as being one pathway that's specifically important in the CRF neurons in the central amygdala for consolidating extinction memories or inhibiting extinction memories. with it through its downstream transcriptional pathways. More recently, Joe Ledoux's work really outlined the physiology of consolidation in the amygdala, showing that LTP, that tone shock pairing in the rat amygdala essentially led to a LTP-like associative process between the medial geniculate thalamus and the amygdala. And we now know that there's a whole host of LTP-like physiological components that are ongoing in the amygdala during memory consolidation, and particularly threat consolidation. My work started with Mike Davis, really looking at some of the early molecular components within the amygdala, and we found a lot of different gene pathways in our early work that were changed in a dynamic patterns that looked very much like Jim McGaw's early hypothetical patterns. In the aftermath of fear memory formation or threat shock formation, we found immediate early genes that were rapidly increased and then normally and then downregulated again. These are genes across the amygdala, entorhinal cortex, and hippocampus. Others that were slower to come on and then stayed for longer, and these included a number of more structural genes and BDNF and other plasticity genes, and then some that went down with a similar time course, and these seem to disinhibit circuit pathway. So starting to be able to make sense at a molecular consolidation level on a similar time course. So that and work of many others, Alcino Silva's group and many others with the creb pathways, really have led the field to really thinking about stages of memory encoding following traumatic event consisting of early consolidation, reconsolidation, and extinction. And so, again, the traumatic event can be a whole host of contexts and cues and people that together form a short-term threat memory. And that is then, this engram is then consolidated, of course, over the course of minutes to hours to days. Where every time it's reactivated, there's a working memory formation that then is likely, the terminology that we would use for when that new threat response is occurring. And if the threat response is occurring and there's not sufficient negative data to modify it, it's reconsolidated and possibly strengthened. And some people may be more likely to sensitize this pathway than others, and maybe that's part of that sensitization subset of PTSD. If the memory is reactivated, nothing bad happens, and it's reactivated again and nothing bad happens. Maybe you're telling your family about it and they're supporting you. You're telling a therapist about it and they're having you tell it over and over and over again. Then you're going down the path of extinction, and you're creating new safety memories and new engrams, where now safety memories are helping to be consolidated on top of the prior threat memories and starting to provide more context and places and components where those memories are no longer threatening. So on that background, I'm going to start telling you about some work from our group and others focusing on what's happening in the early phases, days, weeks to months of memory consolidation in humans, and what that tells us about PTSD and how that overlaps with some of our molecular genetic understanding of PTSD. This work in our group started with an initial study, two-site R1 between Emory, where I was up until 5 years ago, and Charlie Nemiroff's group at Miami. We identified people in emergency departments, and we enrolled almost over 500 people in the immediate aftermath of trauma, basically right off of the ambulance in the emergency department as soon as they were medically stabilized. And we were able to get bloods from the time of essentially the first stat pack in the emergency department, as well as a whole host of data we collected in the emergency department and up to 6 months later. And these data looked really promising and showed that we could really could do this in a meaningful way. So that plus a number of years of effort working within IMH led to a large-scale study called the Aurora Study, a multi-site study of post-trauma predictors that really is goal is to be, create the largest data set available to really understand the timeline and components of the human consolidation process in the aftermath of trauma and the long-term trajectories of trauma exposure. And of course, while PTSD is where we started, we know that trauma begets depression, relates to substance abuse, relates to increasing in other psychiatric disorders, and has different levels of functionality. So we're really seeing this as a transdiagnostic data collection. And our goal is to collect data on over 5,000 to 6,000 people total, and they're everything from digital devices, wearables, phone-based apps, cognitive data based on the web, et cetera. So everything we can digitally, very densely across that 2 to 1 year period, as well as getting blood genetic data at the time of enrollment. And then a subset of about 800 people we're bringing back at 2 weeks and 6 months to get much deeper phenotype data, neuroimaging, neurophysiology, startle physiology, and additional blood-based data on. So some of the early data from these kinds of studies suggest that as a field, we're on the right track. So this is work from Jenny Stevens, now an assistant professor formerly from our group at Emory. And what she showed was that if we look at people's, so first of all, overall PTSD symptoms in figure A here, that we have a whole range of people whose PTSD symptoms, at 2 to 4 weeks, so about a month, I guess at the latest. This is a full range. Some people with very high PTSD symptoms, all of this in the clinical range. Some people with low symptoms and not particularly bothered. If you follow these people for a year, some people are low and stay low, some are high and stay high, some get better, a few get worse. And if we then look at their amygdala activation to fearful cues, so this is a specific task-based fMRI for this particular figure, in which they're looking at fearful versus neutral faces and simply asking amygdala activation. And it turns out that amygdala activation at 2 weeks after the trauma, so the time we would never call PTSD clinically, it would be a huge stress, and many people do recover after that period, that predicts quite robustly the 3-month PTSD symptom and the 12-month PTSD symptom, and also dorsal anterior cingulate, which is highly co-linear often with amygdala activation. So usually the DACC and amygdala go together in terms of increased arousal, increased threat in PTSD literature. Momo done a lot of this work, whereas the hippocampus and the ventral medial prefrontal down here go in the opposite direction. Anyway, so it looks like amygdala and ACC are positively correlated with later PTSD development in a predictive fashion. On the flip side, Sana Van Rooij, also an assistant professor at Emory, focused more on hippocampus and on other kinds of functioning. And this was a task looking at a response inhibition task that requires hippocampal and cortical. And she nicely showed the opposite, that higher hippocampal functioning, so in this case, further on the y-axis, was associated with much less PTSD symptoms. And that's, again, consistent with higher hippocampal functioning is associated with more contextual encoding. And we know that hippocampus is also involved with more extinction responding and the contextual basis of extinction responding. So that people at 2 weeks with higher hippocampal functioning inhibitory behavioral task showed much less PTSD symptoms at both 3 months and 6 months. And interestingly, if you do a multiple regression, an elastic net regression, hippocampal activation at 3 months was predicted, but so were other things you would predict, race and socioeconomic status and pain. Once we got to 6 months, hippocampal activation was by far the strongest predictor, which is quite interesting. And then in terms of devices or tools or biomarkers that we might be able to use early in the aftermath of trauma, we went back to an oldie but goodie galvanic skin response. And so we were able to use a program called eSense, which provides a very low-cost way of doing galvanic skin response that can be attached to an iPhone or an iPad, and do it in the ER sometimes while people were still on their attached to the bed or the stretcher. Anyway, what we found was that in the ER, so again, hours after trauma, when they're telling us what brought them into the hospital, their galvanic skin response correlated very robustly to their PTSD symptoms 6 months later. And there's positive versus negative, suggesting that measure of adrenergic sympathetic arousal in the immediate aftermath of trauma may still be a very important early predictor of long-term trauma. And these are all things that we could think about combining with early prevention studies to be able to look at in terms of, can we move the needle on early galvanic skin response? Can we move the needle on early amygdala activation, or help the hippocampus be active sooner? All of those might be involved in moving the needle on early consolidation in humans with potential PTSD. So how do some of these data map onto where we are with human biology and human genetics data? So again, there's been multiple waves of genetics, and we're now very much in the large GWAS era. The GWAS of PTSD was a few years behind depression and PTSD, but we're rapidly catching up. So in 2018, we had about 20,000 total in our multi-site psychiatric genomic consortia. Now the PTSD Psychiatric Genomic Consortium, which is led by Caroline Nevergelt, Kirsten Cohn, and Murray Stein, and myself, has over 200,000 samples. And the most recent large-scale publication, the Million Veterans Program, which is now also part of the broader PGC, had multiple hits, and I think the latest count is we're close to about 20 different large-scale genome-wide association hits associated with PTSD. So I think the time is coming soon where we'll really be able to have large-scale genetic drivers of those who are at risk for PTSD to start mapping back onto our circuits that we really are beginning to understand quite well. These are the data from some of the early studies, confirmed heritability, and that twin study heritability in PTSD is similar to twin study heritability in schizophrenia and bipolar for people who've been exposed to trauma. And an interesting finding for which we don't yet understand is there seems to be more heritability in females to males. Happy to discuss that. The reason I have this particular slide up and trying to bring these fields together is while there are many different hits, and as with all GWAS studies, there are many genes that we don't yet know a lot about, or gene deserts, there are starting to be some in really interesting areas. And one of the top hits in chromosome 17 is in a cluster of genes here. So this is, again, each dot is a single polymorphism with the inverse log P, and 10 to the -8 is roughly genome-wide significant. And this cluster of hits, this locus, includes the corticotropin-releasing hormone CRHR1. And the reason I think the field is so excited about this. Obviously, what we don't want to do is go immediately step backwards from GWAS and say, okay, we threw out candidate genes so that we could do GWAS and learn new pathways. And as soon as now we find something that maps an old candidate, we jump back onto it. But I think it is really critical that it does help us to validate at least some of the biological stories that the field understands well, and for which there's a huge amount of data. Similarly, the dopamine 2 receptor, came up on schizophrenia. It wasn't until number 150 something in the GWAS, but at least there's some validation that dopamine pathways, independently in a fully unbiased genetic way, are also associated with psychosis. So again, the hypothalamic pituitary adrenal axis is really one of the best understood components of the stress response. The amygdala and BNST project CRH neurons onto the hypothalamus. CRH is released into the pituitary, leading to ACTH release, leading to cortisol release. And there's a lot of data on altered and dysregulated cortisol feedback on these systems in CRH in post-traumatic stress disorder. From our own group and others, again, long predating that new GWAS finding, had shown that CRH polymorphisms, so again, polymorphisms in the same receptor, were associated with differential amygdala activation and with differential sensitivity to stress-related disorders like depression. Genes that were upstream of CRH, for example, the PACAP receptor is upstream of CRH and CRH receptor, and that's been very much associated in a number of genetic studies with PTSD, as well as differential amygdala activation with PTSD, depending on SNP polymorphisms, and interestingly, potentially different sex differences based on estrogen regulation of the PACAP receptor gene. And then finally, one of the best understood gene by environment stories is FKBP5, which is a chaperone that interacts with the glucocorticoid receptor and really dials up or dials down the glucocorticoid response that regulates CRH in the feedback loop. And that differential FKBP5 functions associated with differential PTSD symptoms as a function of childhood trauma and differential amygdala activation. So these are things which really were holdovers from the pre-GWAS era, but are still have continued to be replicated and interesting, and are all part of the broader story. I think of the CRH pathways, both neurally and molecularly, seem to still be robust in these disorders. At a rodent and circuit level, again, there's been much work on the CRH and CRF pathways. This is early work I did at Emory with Charlie Nimroff's group, showing that if we used a genetically modified virus to overexpress CRF using an endogenous CRF promoter, in the CRF neurons, we would get an enhanced startle response and an enhanced fear response, similar to what we see in patients with PTSD, measuring startle and fear responding. if we physiologically overactivated the CRH neurons, in this case, we did that by knocking out a GABA receptor, specifically the alpha-1 GABA-A, that is involved in modulating many of these cell populations, such that if we knock out the GABA-A alpha-1, we presumably have a hyperexcitable set of cells. And when we did that, and specifically the CRF neurons, again, we could show that that increased CRF expression did not alter the initial fear acquisition process during tone shock pairings. But then when we measured fear expression and fear extinction, animals that had a GABA knockout on the CRF neurons had higher levels of fear over time, again, consistent with either an enhanced initial consolidation or a deficit in expression and extinction. And they also had increased HPA axis sensitivity. So just a tidbit of the data suggesting, and for which there's hundreds more papers showing CRF regulation of stress and PTSD-like models in animals. Of course, where we are now as a field is really focusing much more deeply on mechanisms and microcircuits. And so whereas we used to talk about the amygdala, now we know there's 10 to 15 different subnuclei of the amygdala, and instead of excitatory and inhibitory neurons, we have many different subtypes of cells, including differential subsets of excitatory and inhibitory neurons within each of these regions. And I'll dive through some of these subsets in different ways through the rest of the talk, but wanted to talk about specifically for a moment the CRF specific subpopulation in the central outer amygdala. Again, some of this work were things we were working on historically anyway, but have particularly, I think, new significance and interest now that we, have the potential of this genetic locus in multiple GWAS studies of PTSD. So in this particular line, there's 3 or 4 different CRF promoter lines now. This is one of the CRF virus pre-promoter lines. We had made one a number of years ago. Tali Barham has made some. Our question was, we wanted to dive in a little bit deeper and say, can we understand what are the gene pathways that are differentially regulated, specifically within the CRF, and I'm going to flip back and forth, I apologize, corticotropin-releasing factor, CRF, corticotropin-releasing hormone, CRH, same gene, same protein, a field that's never quite landed on what it's going to call it. For this paper, we called it CRH. So we used the CRH creep driver line crossed with the TRAP, ROSA6, FOXP, GFP, L10A line. Briefly, this was created by Matt Heinz's group and Miriam Hyman, who's now at MIT, and Robbie Finster, who's a junior faculty member in our group, was one of Miriam's students. What this does is essentially add a GFP link to ribosomes that are actively translating proteins, so that you essentially, and then you pull down you can do a punch, so you can essentially do a biochemical level extraction, but through the co-IP pull down, only extract RNAs from the cells that are expressing the tagged ribosomes to essentially get a cell type specific, but a reasonable amount of RNA. So this is before we started this, before some of the new single cell seq tools that I'll talk about at the end were available. And we did this then in several cohorts of animals that were control, animals that had fear conditioning, and animals that had extinction. And we did it right after 2 hours after fear conditioning or 2 hours after extinction to look at what genes were changed in the CRF central amygdala neurons during the consolidation window of either fear consolidation or extinction, and found a whole set of genes. And the take-home message is that particularly in these CRF neurons, we found creb differentially regulated and particularly downstream genes of creb in very interesting ways. So the first finding that we were disappointed by, but we thought it was interesting when all was said and done, is that when we looked at the consolidation window of fear extinction, we found a whole host of genes that were differentially expressed at experiment-wide, gene-wide differential level cutoff. So these all survived multiple corrections. In the fear expression versus tone alone, so we had 3 groups: tone alone, fear expression, fear extinction, comparing to tone alone. The fear expression consolidation group, found a lot of genes that were nominally significant, but none that survived whole genome significance. And we think this is the reason is when we started looking back at the literature. So Larry Zweifel and Richard Palmer at Washington and others have done more recent work on the CRF neurons as well. And their data suggests that the CRF neurons may be particularly involved in sort of moderate to weak threats and not significant threats. And then they may be involved in extinction. And that looks like exactly what we're seeing. We're seeing a set of genes that seem much more actively, dynamically regulated by extinction. And in a separate follow-up, we found that CRH gene itself was activated actually by both tone exposure and fear conditioning, compared to other genes that we did not expect to be changed or went down, suggesting that a new tone alone in a new context may be sufficiently threatening or novel that for these cells, it doesn't even matter. So that a little bit of novelty and a lot of novelty may turn these cells on no matter what. Whereas where you really get your meat of what the role of these cells are doing is expectancy of fear and then it not happening, and that's where you activate the extinction process. So that's at least one way of thinking about Zweifel's data and ours, in that we're not seeing much signal in these in the fear encoding itself, but in the extinction encoding. So after seeing a whole host of genes, again, that were downregulated, that were immediate early genes, that were kinases and other genes, many of which were all within a transcriptional unit that was downstream of creb. So we asked, what happens if we overexpress creb or knock down creb? And basically the take-home message is if we overexpress creb, we can push the behavior in the opposite direction. So we used a virus from Rachel Navy to overexpress, Cree-dependent virus overexpress creb only in the CRF neurons. And when we did that, we don't find any effect on plusmates or open field against sort of standard anxiety-like behaviors, and we didn't find any effect on locomotion, so there was no broad effect. We also found what appeared to be no effect on at least the early steps of fear acquisition. But when we looked at fear consolidation, we found significant increase initially in fear consolidation and an ongoing deficit in fear expression. And then when we tested the animals again to look at the expression of fear extinction from this within session extinction experiment, the crab animals also showed more fear. So what it looks like to us is that in the CRF cells, if you're driving synaptic plasticity pathways, and if we think about Sheena Jocelyn's crab capture sort of models, you're driving the CRF cells to more likely be in a state of crab capture, you'll see perhaps more fear consolidation, but definitely more fear expression and a deficit in fear extinction. And that what the normative response seems to be during extinction is that the creb pathways are all downregulated. And so normal downregulation of creb sensitive pathways in the CRF neurons allows the consolidation of extinction to occur in a way that it doesn't with consolidation. So that was one new way also to bring back our understanding of CRF in these threat sensitive pathways with some old friends as well. So wanted to now move into the last section and talk about other pathways that one might could target in our understanding of bringing together the molecular and neurocircuit models of fear consolidation and fear processing to think about possible interventions. So while I'm not going to talk about many of these, kind of where the field was a few years ago is, okay, let's look at, this is a consensus platform, and these are all the different gene pathways are really pharmacological targets. NMDA receptors, cannabinoid receptors, glucocorticoid receptors. So again, dexamethasone or hydrocortisone would fit there. So there's some interesting things to try, very few of which really grow out of our full understanding. Though the cannabinoid modulators and phi inhibitors are quite interesting. How do we identify novel tools and novel pathways? So again, we've been doing this for a number of years now, looking at what are the gene pathways differentially regulated during fear consolidation. And tachykinin 2 is one that we've known about for a while, but has some new data that I'll show briefly. So again, tachykinin 2, and we did some early bulk gene expression experiments, was a gene that in the central amygdala is expressed highly in a dynamic acquisition-dependent way. And it's expressed with associative learning and not stress alone. It's expressed in the centromedial neurons, specifically in a population that's complementary to enkephalin or PKC delta in the centrolateral population. So it's expressed in a learning dependent way. It's expressed in the output neuronal subgroup of the central amygdala. And it seems to be involved in fear consolidation. So there's an antagonist of sanitation that was already available. If we give a sanitation either 30 minutes before, 10 minutes after, 1 hour after fear conditioning, but not 4 hours after fear conditioning or 24, we see a nice, robust decrease in fear memory consolidation when tested the next day. And when we do that osanetan blockade in fear consolidation, we also see decreases in molecular evidence of fear processing. For example, PACAP receptor I alluded to earlier is normally very highly increasing its expression in the amygdala with fear consolidation, but if osanetan blocks fear consolidation, you don't see that increase. If we look at this more specifically, so the first consolidation experiments were all systemic. This is a cannula experiment in mice, again, giving osanetan, the TAC2 NKB antagonist directly in the central amygdala. After tone shock pairings, then we test the animals the next day, and we see a robust deficit in fear in the animals that receive the drug. If we look at this genetically, we can overexpress tachykinin 2 with the lentivirus, specifically in this general central amygdala area. When we overexpress it, we increase the fear expression. So we can push fear consolidation up or down based on inhibiting or overexpressing tachykinin 2. And when we did a final replication combined experiment, we could show that we could push the memory in both ways. So again, this is 24 hours after fear consolidation. And we previously, with the subset, either gave animals GFP versus TAC2 overexpressing lentivirus, and then immediately after fear consolidation, during the consolidation window, we gave drug versus vehicle. In the control GFP group, if we give the drug to block tachykinin pathway, we get a decrease in consolidation when tested 24 hours later. If we give the virus alone to overexpress tachykinin, we replicate the original finding and get an increase in consolidation. If we give the virus to overexpress it and give a sanitation to block the downstream effects, we have no effect on consolidation. So it looks like we can push and pull this in both ways. More recent work by Raul Landero, formerly from our group now in Barcelona, along with Tigrani and others, has shown that that tachykinin 2 cells specifically are can respond in an increased way to fear consolidation, and that if we optogenetically activate the tachykinin 2 expressor cells, we get an increase in consolidation. So this is with channel rhodopsin, activating with channel rhodopsin tachykinin 2 positive cells during acquisition of fear, and then testing them 2 days later. And again, in the black bars here, you see increased fear. So we've been able to show an increase the tachykinin role in consolidation, both with agonism, overexpression of the gene, and activation of the cells. And more recently as well, Muriel Zelikowski, who's now at Utah, and David Anderson's group while at Caltech, showed that tachykinin is also involved, particularly in the medial amygdala, but also some from the central in chronic stress isolation and in aggression responding. So it seems like an interesting pathway in many different ways. I wanted to wrap up here with one final set of experiments that we've been working on for quite a while. And this is another subset of cells in the basolateral amygdala marked by the Phi1 marker. And it's a subset of maybe 30 to 50% of the CAMP kinase 2 positive cells in the basolateral amygdala. What's interesting is this subset projects to the accumbens and not to the central amygdala. So it seems like this is the accumbens reward path of the BLA and not the central amygdala fear path. And also, we also found early on with Zarin Jasnow and more recently with Ken McCullough, that if we activate these cells, either with channel rhodopsin during fear acquisition, we can block fear memory formation, or we halo rhodopsin, we inhibit the cells, we increase fear consolidation. So these cells seem to have an opposing role to fear consolidation, and separately, they seem to enhance some level of repetitive behavior in acquisition. When Kenneth did a cell sequencing experiment doing doing fact sorting to find genes that were specifically expressed in Thy1 cells. He found a whole bunch of interesting ones, some of which have come up before. RSPO2 come out of Josh Kim and Tony Galba's lab in a similar pathway. One of the genes that was very interesting to us was the Neurotensin II receptor. When we targeted, and the idea is this is a GS activating receptor. We've never thought about it before, though we know neurotensin is involved in emotion, expressed in the Thy1 cells. If we then give an off-the-shelf agonist of the Neurotensin II receptor to these animals to activate the cells that we think are fear-off cells, and we do that during acquisition of fear and then test them during fear expression, we see a profound blockade of fear consolidation here in the red line, suggesting that this is a rational therapeutic that can work. We can start to identify receptor targets in these very specific pathways that actually may give us new therapies for understanding how we can block fear consolidation. So then in summary, the field has a number of already existing over the last 5 or so years types of drugs that are already available that potentially based on our circuitry in terms of hydrocortisone modulating CRF, neurotensin agonists modulating the Thy1s. I didn't talk about the Aurora 2A antagonists, but those are also fearon cells and the tachykinin cells, all of which that potentially could block fear consolidation in the immediate aftermath of trauma. And so we're already starting to think about, could we take this Aurora study that we've now, got close to 5,000 people on, and in the next generation of that study, start doing interventional studies to test some of these ideas. The future of the field, I didn't show much data today yet, but is using single cell sequencing across species and really being able to use the mouse, not so much as a behavioral model for a human, but really as the brain model, and say, if we understand X, Y, and Z specific cell population of fear-on or fear-off basolateral neurons in the mouse, do they exist in the human? So the same receptor subtypes exist in those cells. And if we're going to start working on understanding specific microcircuit targets, do we know those are going to be convergent in the human as well? This work overall has a number of implications for post-trauma intervention, focusing on molecular consolidation, circuit consolidation, and systemic consolidation. And I think as a field, we've now got many ideas, and so it's really how we start taking all these ideas in a funnel and really understanding which of them have the best chances and how as a field can we really target this problem. And I think through that process, the understanding of biomarkers of trauma response and recovery, whether it's through the human condition and the natural experiments like, unfortunately, trauma and PTSD, along with animal-based studies where we really understand through 100 years of work, really the circuits underlying threat and fear-based memory consolidation, are we finally to the point that we can start to bring these together and really have a neurobiologically driven precision medicine approach to better intervening and understanding psychiatric disorders based on fear and threat. Thanks so much to my team. I've tried to thank everybody as we went through. Thanks to NIH and for all of our support over the years. Thanks so much for your attention today.